Hello everyone, my name is Nigar and I am hematology teacher in Baku State University. Today we are going to be more familiar with the processes of measurement hemoglobin concentration, red blood cells count and packed cell volume. Usually all these three experiments were carried separately, but now with the help of Sysmex XS500i it's possible to carry all these three tests in one run. Here you can see the Sysmex itself. So, the XS series is a great addition to Sysmex X class hematology analyzers, and despite being the smallest, its quality is undiminished. All the XS series analyzers are specifically crafted for laboratories that, despite the smaller throughputs, still need to deliver detailed, reliable, and highly accurate results. Before carrying out the experiment, I'd like to introduce XS500i to you. So here you can see its software and the apparatus itself. Most important thing that I have to mention here, it's needle that will suck the blood out of the sample. Here we have our blood sample. We have to fill in the blood till the black square on the tube. And also, before carrying the experiment, you have to mix it, but quite gently, in order to not damage the cells. Now, our sample is ready to be presented. We put the sample till the end and press the button over here. As you can see, there is no needle here. That means that XS500i sucked the blood out of the sample. And now it's ready to be presented on the computer. So the next step is working with the software itself. Here you can see manual. You press the manual. Here we can see the opening window named manual sample number XS. So here we fill in all the appropriate information related to our sample, starting with the patient ID, ending with the last name, sex, etc. So here we can see the patient's first name, birthday, as I said. Also there is two options in terms of discrete and capillary modes. We opt for the right and appropriate for us, and then press OK. So now we're ready to get the results. Here we can see Sample Explorer. We press the Sample Explorer. In the opening window we can see the list of samples that we were working with, but as you remember, we had sample number 2, we press it, and now we get the results. So the last but not the least thing is results interpretation. As you remember, our goal was to measure hemoglobin concentration, to know red blood cells count and the packed cell volume. All the three measurements in a row we can see over here, but other thing that our XS500i is happy to present is other quite large variety of measurements. Well, it uses the same globally renowned detection technologies as Sysmex large analyzers, and thanks to the absolute counting principle, you don't need to calibrate the particle counts yourself, which will save you significant amount of time and effort. Thus we became more familiar with the data that main menu presented to us. I also want to point out another menus and information that they have. So it's a graph menu. It's a cumulative menu. Here you can see you can put for CBC, it could be numerical, graph, distribution. It's Q flags menu, service menu, where again you can see there's so many options over here, and also research menu. Well, this is all the results that we have. I wish you good luck with your studies, and stay healthy. Bye bye.